My project is on penicillin. Penicillin comes from Penicillium mold. The mold produces penicillin, which prevents bacteria from being able to reconstruct their cell wall. This causes the bacteria to rupture and die. However, penicillin does not affect humans because our cells do not have cell walls. Due to these properties, penicillin has been utilized as an antibiotic to help fight infections. This is an overview of how penicillin is made. Firstly, freeze-dried spores are mixed with an agar multiple times. Then it is placed into a fermenter for about a week to cultivate. The aqueous solution of the penicillin is then separated from the solid mold. The product is then cooled to release the heat that is produced from the mold, and then it is put into a holding tank to start the purification process. After purification of penicillin, the product is then dried and ready to use. During fermentation, penicillium is mixed with an agar in a tank, which can include glucose, corn steep liquor, and some acids and bases. Since the mold is aerobic, the tank has a valve to add oxygen into the tank. It also has a valve to output air pressure from the gases the mold releases. There is also a heating and cooling valve in order to keep the temperature between 23 and 28 degrees Celsius. A motor is used to mix all of these components to keep the mixture uniform. The pH is kept at around 6.5. In order to remove the penicillin from the mold, the penicillin needs to dissolve and become aqueous. By adding amyl acetate to the filtering solution, penicillin dissolves and is effectively separated from the mold. To separate the penicillin part from the solution, centrifugal extraction is performed, where the penicillin rises to the top and the solution drops to the bottom. The penicillin part is removed for further purification. Penicillin is purified by continuously adding acids and bases to further separate the impurities out of the penicillin. Some of these acids and bases are chloroform and ether solution. Phosphate buffer is added to keep the pH at 2 to 3. This process is done at around 0 to 3 degrees Celsius to keep the penicillin from degrading, for it is not stable at higher temperatures. After the penicillin has gone through multiple stages of purification, sodium bicarbonate is then added in order to precipitate the penicillin into a powdered salt. With the penicillin now a solid, it is easily removed from the aqueous solution through basket centrifugation. The powder is then dried through fluid bed drying. On the bottom right is the base of penicillin, which is 6-amino-penicillinic acid, or 6-APA. Different types of penicillins are made through a change in the R groups on the amine group, which can be shown on the top left-hand corner of the 6-APA molecule. Now on the left side of the screen, you can see a bunch of different R groups. These R groups attach to the amine group of the penicillin base in order to make different penicillins. The first two penicillins, penicillin G and penicillin B, are directly derived from the mold. All of the penicillins below it are synthesized in the lab. This is an example of how ampicillin is synthesized in the lab. In this example, benzyl chloroformate reacts with phenylglycine, the R group, to produce an intermediate. The bond on benzyl chloroformate gives an extra electron to chlorine, becoming electrophilic, and the bond on phenylglycine gives the electron to the oxygen, causing the molecule to become nucleophilic. This causes the two molecules to come together and form a new molecule with hydrochloric acid as a byproduct. The newly formed intermediate then reacts with ethyl chloroformate in the presence of triethylamine. The reaction also produces another hydrochloric acid and binds at the carboxyl group. This new intermediate reacts readily with 6-APA in the presence of sodium bicarbonate. This releases lactic acid and forms a sodium salt penicillin with a protected carbonyl group. The giant molecule then goes through hydrolysis, removing the protected carbonyl group, resulting in ampicillin. These are my references, and thank you for watching.